Hello, my name is Susana. I'm Bachelor in Computer Science from the University of London. Today, I'm presenting my research project developed during the Research Fellowship Program at Biotech. The title of the research is Identification of Novel Therapeutic Targets in Psoriasis Skin, an isoform approach. Psoriasis is an autoimmune disease mainly characterized by sustained skin inflammation, abnormal keratinocyte proliferation and differentiation, decreased apoptosis, vascular abnormalities, and dermal inflammatory infiltrates. Histologic changes in psoriatic skin include parakeratosis and an increased number of vascular structures. The patients usually present lesions in portions of the skin, but in extreme and usually life-threatening cases, 100% of the skin could be involved. This characteristic allows to compare lesion and non-lesioned skin from the same psoriatic patient. Although the skin profile is the hallmark of the disease, there is evidence of its systemic impact and increased risk of developing other diseases like cardiometabolic disease or chronic kidney disease. NGS transcriptomic psoriasis dataset have been analyzed at the gene level and have unveiled relevant inflammatory pathways, but current treatments modulating the immune response have important side effects and they do not cure. Some widely used psoriatic drugs include TNF-alpha inhibitors that can effectively clear lesions but may present safety concerns like increased serious infection risk. Since psoriasis is a chronic disease, patients usually require long-term treatments, which could increase the risk of presenting side effects. Hence, alternative targets for treatments are required either to reduce the side effects or support a cure. The objective of this study is to identify such new treatment targets from an isoform level perspective. This project used this public RNA sequence dataset, which originally contains 147 samples of 5 conditions. It includes atopic dermatitis patients, psoriasis patients, and healthy controls. For the purposes of this study, only samples from control and psoriasis patients were selected. The skin transcriptomes of 45 subjects were analyzed at the isoform level. From the transcriptomic pair reads expression, isoform levels were generated by running the pipeline shown at the top left of this slide. This pipeline generated 2 main output expression levels files for isoforms, an FPKM table and a raw read counts table, but tables were split into 3 groups for further comparison. Here are the groups selected, which corresponds to the healthy control versus the psoriatic non-lesion, healthy control versus the psoriatic lesion, and psoriatic non-lesion versus psoriatic lesion. The output table containing FPKM read counts isoform expression levels underwent log transformation and quantile normalization with a threshold of 5. This table was used to conduct PCA exploratory analysis and remove outliers from the samples. In this slide, the resulting PCA plots after removing outliers are shown at the bottom, and a clear separation between conditions can be observed. Once outliers were identified, the respective samples were also removed from the 3 tables containing raw read counts, which were previously prepared by removing genes with a sum less than or equal to 5 across all samples. Each prepared isoform raw read counts expression table corresponding to each of the 3 groups was uploaded for differential expression analysis. At this step, HR was used with the parameters log fold change less to -1.5, p-value less than 0.01, and false discovery rate less than 0.05. For the pairs of compared conditions which are identified, for healthy versus non-lesion, a total of 107 transcripts and 49 new transcripts, novel isoforms. The second group, healthy versus lesion, identified 4,819 total transcripts. For the third group, it identified a total of 2,721 transcripts. The top 10 downregulated and upregulated isoforms were selected for each group, and the resulting sets were compared between conditions to extract the markers of psoriatic skin and the markers of lesion psoriatic skin. This slide presents the matched isoforms. Among the differentially expressed isoforms, the novel sequence corresponding to filamin B was further analyzed. This isoform was selected because its respective gene is not identified at gene-level studies, and the product of the gene influences molecules and mechanisms related to psoriasis. For example, MMP9, VEGF-A, cell proliferation, apoptosis, RAS-ERK pathway, and angiogenesis. Here are shown the other identified top downregulated or upregulated isoforms. Some of them are already identified as differentially expressed at the gene level. To select the nearest known transcripts, the novel sequence was used for multiple sequence alignment. This figure shows the novel isoform aligned against the 28 known isoforms available at its sample. The sequence of the novel transcript was further compared against the nearest identified isoforms, which are the canonical sequence, the isoform 8, the isoform 9, and their coding sequences allow us to predict that the translated protein of the new sequences may be the same that the filamin B isoform 8. The following figure represents the differences in the sequences during the comparison. Filamin B isoform 8 has an extra amino acid sequence when compared to the canonical one, but it maintains the associated biological processes. The novel transcript has that insertion coding for the extra amino acid sequence, but also contains non-translated nucleotide sequences present in the canonical sequence and in the isoform 9. Considering the predicted translated protein, the deficiency of the novel identified isoform is also expected to decrease apoptosis, increase cell proliferation, increase cell invasion mediated by RAS-ERK, and lead to higher MMP9 and VEGF-A activity. All of them are characteristics of psoriatic lesion skin. The deficiencies of the filamin B are listed here. Many of them match the psoriatic lesion profile skin. And here are the relation of such molecules with psoriasis. So within this context, filamin B might represent a potential target for future treatments, and it might provide an alternative option for regulating MMP9, VEGF-A, and RAS-ERK pathways. Unlike MMP inhibitors, filamin B will not block other MMPs, so this could decrease side effects. At the same time, the activity of VEGF-A could be regulated, so it could lead to reduced formation of capillary structures. Isoforms of genes RAV14, S100A7A, DCN1, SPRR2F, and DERB4A were found to be representative of the psoriatic skin profile. The first ones have been found to be differentially expressed in psoriasis at the gene level, all of them also upregulated. However, when comparing psoriasis lesion versus the control, TCN1 expressed 3 differentially expressed isoforms, whereas for non-lesional versus healthy controls, there was only 1 differentially expressed isoforms. Hence, one isoform, which corresponds to the canonical sequence, can be an indicator of psoriatic skin in general, while the other 2 TCN1 isoforms, which are not translated into a protein, might be relevant in flare-ups. On the contrary, FRAP14 is not identified to be differentially expressed at the gene level, and its novel transcript found in this study is predicted to produce the same protein as the canonical sequence. Isoforms of genes EA of 4A2, filamin B, CACNA1H, CAST-ADD3, and RER1 are proposed to be indicators of lesion psoriatic skin, and only CACNA1H is already known to be differentially downregulated at the gene level. The downregulated isoforms of CAST are thought to be involved in muscle protein degradation in living tissue. Furthermore, CAST reduces levels of protease calpain, a protein that presents increased levels in arthritis rheumatoid and other autoimmune disease. For ADD3, one of the 3 differentially expressed isoforms is expected to impact the cytoskeleton organization, while another transcript does not code for a protein, and the other one represents a novel one. A novel transcript of filamin B was found, whose translated protein is predicted to be the same as UniProt identifier for the isoform 8. Its downregulation is expected to increase the activity of MMP9 and VEGF-A and the proliferation rates of keratinocytes. The expression and activity of MMP9 and VEGF-A are directly proportional to the severity of the psoriatic lesions, and its upregulation can produce psoriatic-like lesions in models. However, current drugs targeting them have severe side effects, since both molecules can be regulated by filamin B. This study proposes that filamin B supplementation or treatments aiming to upregulate this gene could be effective in flare-ups clearance. I would like to thank Dr. Raghavendran and Mr. Ilya Brodsky for mentoring this project, and also thank you for Sonalika Ray for all the support and all the team from Biotech and OmicsLogic. Thank you so much.